So the plan for today is to go through the evolutionary mechanisms and finish that up. Again, a reminder, discussion sessions start this week, so perhaps you've already had your discussion session. If not, you should be having it over the next 2 days. I wanted to alert you, I have a conflict with office hours tomorrow. Normally my office hours are Thursday at 2. I apologize for any inconvenience this may cause, but I can't make this one, so I've rescheduled. That's just this week only. Normally it'll be Thursday at 2, but this week only it'll be Friday at 4, which might even work better for some of you, I hope. Same location, 5248 McGaw Hall. Perhaps I'll see some of you there. So we've been calculating allele frequencies, genotype frequencies, phenotype frequencies, using Hardy-Weinberg, learning how to use Hardy-Weinberg. So we're going to go through another example or application today to have an opportunity to practice on using our ability to calculate frequencies. And to do this, we're going to use some data that are in table 26.1, which is in front of you right now. What this table shows is some of the genotype frequencies for one particular gene in humans that affects blood type. This may be an unfamiliar blood type to some of you who are familiar with some of the other kinds of blood types. Blood is really complicated, and there are many, many different ways to type. This specific way is a single gene that codes for protein on the surface of red blood cells. There is a so-called M version of the protein and an N version of the protein. So it's just one gene, primarily two alleles, very simple case. And the scientists who are interested in investigating the distribution of these different genotypes in human populations went to a number of different locations and took a lot of blood samples. The locations are shown here in the far left-hand column. It shows that the populations were humans in Greenland, two different populations in the United States, Native Americans and Caucasians, an Australian population, and then a Japanese population. I know it's a little bit hard to read this table from where you are, and so I'm going to take this bit right here and zoom in. So that's what we're looking at now. It's just that little tiny bit of the table, which is now a bit more magnified, so hopefully you can see it more easily. And so you can remember, although the locations, the geographical locations for these 5 different samples are no longer visible, you should keep in mind these are 5 completely different samples. So what are we looking at here? Let's start with observed. So this is the actual data that were taken by the scientists who were going to these 5 different locations and taking blood samples from a number of individuals. So you take a blood sample from an individual, then you have their blood in a little vial. You can add the antibodies to M and N, and then see what results you get. And if the blood showed that there was only M there in the blood sample, then the scientists knew that this was an MM genotype. If it was only N, then they would know it was an NN genotype. And if both were present in a blood sample from a single individual human, then it was MN. And what you should notice first is that these numbers are really quite different in the different populations, right? We've got 0.835. Oh, what does that stand for, by the way? That's a decimal representation instead of a percentage, right? We can also think of this particular population, which I believe was Greenland. Yes, it is. 83.5% of the individuals who had blood drawn in Greenland turned out to be MM, right? So this is written as a percent instead of the decimal. 15.6% were heterozygotes, and slightly less than 1%, right, 0.9%, were NN. Now, the data are reported as in percentages of the population from whom the blood was drawn, which was a large number of individuals. So if we look at just the MMs in the different populations, you can see that these differ quite a lot. We've got 83%, 60%, 29%, 2.5%, and 18%. So very different in the different populations. And so there was some interest in, is there any evidence of different kinds of evolution going on in these different groups? And so one of the ways that one can look for evidence of evolutionary mechanisms, of course, is to compare the genotype frequencies that are observed with what would be predicted based on Hardy-Weinberg. So, and of course, for any given row, these have to add up to 100%, right? So if we add these, they add up to 100%, just as if we add the decimal equivalent, then this would add up to 1, right? Does that make sense? Okay, so in order to then turn these observed data into predictions, if in fact the data satisfy Hardy-Weinberg, then the first step is to calculate allele frequencies. So I'll walk through that with you. I'll just keep the very top part of the information on this bit of the table so that to give me room to write. So this is just the very, very top part just for the Greenland data here. So again, here's the observed data, and now you can see the two columns to the right, which are the allele frequencies. And so step number one is to calculate the allele frequencies from the actual alleles that are known to be in the population. So how does one do that? This is slightly different than the examples we've gone through before, where you knew there were so many individuals that were MM and so many individuals. Now we've got proportions. So the math is a teeny bit different, right? So now how do we calculate the frequency of M? Let's define P as our frequency of the M version of the protein, and Q as the frequency of the N. Then if we're going to calculate Hardy-Weinberg, we know that the expected genotype frequency for the MM genotype would be P squared, and this would be 2PQ, and that would be Q squared, right? But where do P and Q even come from? Those have to be calculated, right? So these were measured, and then we can calculate the allele frequencies. How do we do this? Well, let's start with calculating P. So that's the frequency of M, which is P, and so this equals what? We can take this right from the percentages. So there's 0.835 plus half of the heterozygotes, right? So there we go. There's that number right there. Okay. And if you want to try this as a slightly different numerical example, you can pretend that the population has, say, 1,000 individuals in it, and then you would have 835 MMs, 156 heterozygotes, and you could walk through it like that and and calculate frequency that way to go through a slightly different example. But for calculating the frequencies, then, if you have proportions like that, instead of numbers, then you can go straight from the proportions. So if we do this, then we do in fact end up with 0.913, and we can plug that in there. Similarly, the frequency of N, let's actually switch to another color there to make it look different. Pull this out. So then what numbers would we use for this? Well, we need this one, right? 0.009 plus 1/2, again, of the heterozygotes, because half of the oops, that's the wrong place, right? Because half of the heterozygotes are M and half of the alleles in the heterozygotes are N, right? So if we add those together, we get 0.87. One really important point about this is that the allele frequencies that we just calculated have absolutely nothing to do with Hardy-Weinberg predictions, except in terms of we need them to make Hardy-Weinberg predictions. But this P and this N are factual statements about the allele frequency in the data that were collected. They're not hypotheticals, they're not predictions. They're just statements of fact about the data. This is completely different from this next line right here. The frequencies that are expected if we assume Hardy-Weinberg is true. Okay, that is not that is a hypothetical predicted frequency based on certain assumptions, the assumptions of Hardy-Weinberg, right? And so if in fact we take 0.913 and square it and plug it in right here, we get 0.834, for example. And 2 times P and Q is 0.159 and so on. So it does, in fact, turn out that the numbers that we get by plugging them into the Hardy-Weinberg formula very closely match the genotype frequencies that were directly observed. Although the source of the data is the source of the numbers is completely different, right? This top row here, observed, directly measured. This next one, expected, is calculated based on actual P's and Q's. So if we go back to this overall chart then and table, you can see that that's actually true for every single population, even though they're different from each other. In every single case, the expected frequencies are extremely close to the observed frequencies, 0.6, 0.6, 0.29, 0.29, etc. Very, very close. So one could then interpret these results as evidence that there are no evolutionary mechanisms that are non-negligible that are operating at this particular gene in these particular populations at this point in time. It doesn't mean that they weren't earlier, but certainly it means that at this point in time there is no evolution going on at this particular gene, and it also means there's random mating in these populations with respect to this gene. So individuals with a particular genotype are not more or less likely to mate with other individuals. Mating is random. Yes. Oh, front row.
How did they get the expected numbers?
Calculated, right? So does it make sense? So for those of you in the back who didn't hear the question was, how did they get the expected numbers? These are calculated, and they're calculated based on Hardy-Weinberg. So in other words, these are directly observed from the population. Does that part make sense? And then does it make sense that these are just calculated from the data, right? Does that part make sense? Okay, so now we know what P is and we know what Q is. When we have one gene and two alleles, then to calculate the expected or hypothetical genotypes that you would have if in fact Hardy-Weinberg was approximately true, then what you would plug in here for, at this point right here, is P squared. So you take 0.913 and you multiply it by 0.913. And then you put that number in here. And if it's really different, then you say, wow, we got a mismatch here. We may not know which evolutionary mechanism is important here, or even if maybe there's non-random mating with respect to this locus, but something is going on here because it's not actually coming out as if it's random. Does that make sense? Yeah. So yeah, this is straight out of Hardy-Weinberg. So this is 2p, well, this whole one here, 2pq, q squared, and this is p squared. For the just these expected numbers, that's all. Any other questions? Question in the fourth row. I'm sorry, what was that? Row 4. So the 0.178, 0.825, that one?
Yeah. The frequency, we add up, it doesn't equal to 1.
Ah. Good eye. Did everybody catch the question? Right, so if you're quickly adding up all these numbers, then of course P plus Q should equal 1, right? So if they don't equal 1, then something fishy is going on. So either it's a typo, let's see, so this one, right, so 0.825, what is that, 13, yeah. So it looks like we've got sort of an extra here. It might be a rounding error. You know, this might really be 0.8246 or something. I don't know. I don't know if this is a rounding error or if it's let's see, or if it's a slight typo. My guess is it's a rounding error because it's reported to 3 significant digits, right? So that will happen sometimes. In fact, that happened last lecture when we went through an example where if you just take it to 2 significant digits and a third of, if you've got 3 genotypes and each is a third, then to 2 significant digits you've got 0.33, 0.33, 0.33, it only adds up to 0.99. So it's like, where's the missing 0.01? Well, it's just a rounding error. It's a significant digit issue. I suspect that's what that is. Good eye, yes. Absolutely, P plus Q should equal 1, just as if you add all 3 of these, it should equal 1. So when it doesn't, yeah, it's either a typo or a rounding error, and you can usually guess based on the magnitude. My guess is this is a rounding error because it's so close to 1. It's 1.003. Any other questions? Excellent. Okay, so we're now going to walk through the 4 evolutionary mechanisms that were introduced before, one at a time in this particular order. And why do we care about this? Well, understanding the causes and consequences of these different kinds of evolutionary mechanisms make it possible to predict what is going to happen in a particular population, and also to understand what has happened in a population as well. So, as we walk through these, though, we're going to be considering fitness. And so it's really important that you all keep in mind what is meant by fitness, because that's going to show up in every single graph now for the next few slides. So let's go ahead and have a quick test to evaluate, if we're all on board here, understanding what is meant by evolutionary fitness. So here we're doing this as a clicker question. Any response counts. Which of these individuals is considered to be the most evolutionarily fit or has the highest fitness out of these 5? An individual with the biggest muscles, an individual who lives the longest, an individual with the largest brain, the individual with the most offspring, or the individual that's the fastest runner? Not sure? Quick check with your neighbor. Hello, my name is, what do you think? I'll give you 15 seconds or so to chat, then I'll see what you guys think. Okay, everybody's figured it out? Everybody should be clicked in here. Let's see what you think. Okay, let's see if we get 100% here. I can only hope. Fantastic, almost, 98%. Fantastic, I'm so glad. Okay. Thank you. This, of course, is not to say that having bigger muscles or living longer or being a faster runner wouldn't necessarily be adaptive or lead to higher fitness, but just in this list, this is clearly the best answer, because these other attributes don't necessarily lead to higher fitness, although they might. Ultimately, what matters if one is considering fitness is representation genetically in the subsequent generations. So yes, the most offspring. Okay, so now that we're ready to go here, let's go ahead and march through the evolutionary mechanisms. Start with natural selection, which is usually subdivided into subtypes. So let's start with directional selection, which is one of the subtypes of natural selection. And we'll start by acquainting ourselves with the conventions that will be used on the graphs that depict changes in phenotype that result as a result of these different selection regimes. So we're starting out here prior to selection. This is just showing the distribution of a trait prior to selection. What are the axes here? The x-axis is value of a trait, so this is something about the phenotype that you can measure. And of course, if we're talking about selection, then we assume that there is some genetic basis, at least, to this trait. So the value of the trait could be something like height or length of something, number of hairs, fur color, something that one can describe and put on an axis. The y-axis is number of individuals. So the way to read this then, of course, this particular line then goes up and then back down. This is a so-called normal distribution or bell-shaped curve. It's also called a Gaussian. It's got a very characteristic shape. So this is what's being assumed for the distribution of this trait. And so how to read this would be that there's a relatively small number of individuals with this small value of the trait, whatever it is, somewhat more individuals that have this other trait. As we move along the value of the trait, the largest number of individuals of all of these possibilities of the trait values are at the mean right here, right? So this dotted line shows the mean or the average of the trait, and this is where we have the mode or the largest number of individuals that have that trait. And then looking at more exaggerated values of that trait, we have fewer and fewer and fewer and fewer and fewer individuals. Now, we happen to be starting with a normal distribution because many traits do, in fact, show such a distribution where there's particularly large numbers of individuals with an intermediate value for that trait. That's not a necessary part of the whole argument. It's somewhat arbitrary. We could have a completely different distribution pattern and everything else we're saying would be the same. It would just look a little different. Okay? So there's nothing magic about the bell curve. It's just a common distribution pattern observed in nature for traits. So we're starting with this particular distribution pattern in our population. And now selection is going to happen, natural selection. And specifically, you can see that the individuals with the lower values of this particular trait are identified as having lower fitness, whereas the individuals with the larger value of the trait on this x-axis have higher fitness. So the ones on the right are going to have a disproportionate effect on the genetic makeup of the subsequent population. This is actually going to shift the distribution of the trait as shown here in this other panel of the same figure. So after selection, the distribution for the same exact axes is going to look a bit different. And this should make sense to you if in fact the trait has a genetic basis. We've got a shift here. So the mean was at this particular value of x, and now it has shifted over to the right, okay, as a result of this change. So directional selection then has the effect that the average value of a trait is changed. It doesn't have to go in that particular direction. It can go in either direction, but it's a shift and an asymmetry in fitness with a value of the trait as shown here. Okay, if we move on to the next one then, first an example. Here's an example of directional selection. So since this is real data, it looks approximately like a bell-shaped curve, but it's not exactly because it's real data. So the light green shows the distribution pattern prior to some event that took place in this bird population, and the dark green bars show what the distribution pattern was like after this event. And so the x-axis here is body size class, so birds were grouped. So if they were between this size and this size, then they were lumped together into one particular body class, and then slightly larger were grouped together into another body size class, and so on. So notice that before this event occurred in the original population, the mean was around here for a body size of 6 or 7 or so. The y-axis is percentage of birds. You could see it would be fine if it was number of birds or percentage of birds. Either one still makes it a frequency histogram. And then after the event, the mean has been shifted over, so the mean is somewhere over here. So what this means is that the birds that were smaller were less likely to survive or contribute genetically to the subsequent population after this event occurred. So that's an example of what real data look like when directional selection has occurred. And notice that the population size has changed as well. We've gone from almost 3,000 birds to about 1,000. The population size changes are not a necessary part of the argument. What's important is how the distribution pattern shifted. Now we'll move on to the next example, which is stabilizing selection. Again, we're starting with a trait that is normally distributed, as shown here in the top part of the panel, but now we have a different pattern of how fitness relates to the value of the trait. We now have particularly high fitness for those individuals that are right there in the middle in terms of their value of the trait, and individuals on the two extremes are characterized by low fitness. After such selection is operated on this population, then, this tends to squeeze in the distribution pattern as shown here, because the individuals that are either extreme, the particularly low or high values of this particular trait, then are less represented or not represented in the subsequent population. So this has the overall effect then that there is a reduced variation in this trait. The example, and I should point out, notice in this particular case, the mean has not shifted. The mean is at exactly the same point. It's just that the ends have been sort of trimmed off here. There's less variation, less variability in the individuals that have these different traits. The example in your textbook is actually an example from humans. This graph has two different y-axes, so that makes it a little bit harder to read, but let's go ahead and walk through this. The x-axis of the trait is birth weight, so how many pounds was the baby when it was actually born. This is binned into different groups, as shown here by looks like half-pound increments. And so the red bars indicate the percentage of the newborn population. So that's what this red curve right here shows. So what that shows is that most babies that are born, then, and I believe these are US data specifically, are in the range of about 6 or 7 pounds or so. going slightly away from that poundage in either direction, you have fewer babies born of that particular size. And as you get particularly small or particularly large, there's a relatively small number of babies that are born either very, very tiny or very, very large. So that does in fact form a bell-shaped curve as shown here. Now this blue curve here is something quite different. This shows the mortality. The y-axis label is over here on the right-hand side, percentage of mortality. You can notice it is a nonlinear axis. This is to make it a bit easier to see the trend here. And so you can see it dips down and then back up. And so this is the percentage of babies of each size group that end up dying at birth. And so you can see that the individuals that are born right there in the middle are characterized by having the lowest mortality, and mortality increases either as one goes to one extreme or another. The smaller the baby, the more likely it is that the baby is not going to make it, and similarly with a large size as well. And this is because the extremes of either being very, very tiny or very, very large can indicate some sort of problem either with the baby or with the mother. And so that's why this mortality pattern. So this will tend to exert stabilizing selection then on the trait of birth weight of a baby. Disruptive selection is like the opposite of stabilizing selection in terms of the pattern of fitness with respect to the value of the trait. So again, up here at the top, we're looking at a Gaussian curve prior to selection. But now instead of having the highest fitness in the middle and low on the two extremes, we've got the reverse. We've got lowest fitness in the middle and highest in the extremes. So this is the pattern that one sees for traits in which being more extreme is more adaptive. And so when in fact this kind of selection acts on a trait in a population, then this can end up in a bilobed distribution pattern as shown here at the bottom because of excess or extra representation of the extremes in the subsequent generations, and less representation from those individuals in the middle with the in-between phenotype. So this is really quite different. The data that are shown here are for birds and shows an example of birds that survive long enough to breed versus the distribution of beak lengths earlier in this bird population. So the x-axis then is beak length for birds, so shorter beaks over here one extreme, longer beaks over here on the other extreme. Beaks and beak geometry, beak morphology is something that's extensively studied in birds in an evolutionary context because of the very close fit between beak morphology and what kind of consumption, what kind of food items in the natural environment the birds are capable of eating. So birds with very small beaks, for example, find it much easier to go after tiny food that might be harder for birds with larger beaks to pick up, and similarly birds with much larger beaks then are able to crack open foodstuffs that are more difficult for birds with smaller beaks. And so this is a kind of a pattern that one often sees, is that birds with very different beak morphologies then tend to specialize in different food types. And you can see that actually with the orange bars, which show the distribution patterns after the for birds that have survived long enough to breed then. So only the birds that are the more extremes then are surviving in this particular population, which presumably is a function of the food types that are available in the population of these birds. Okay, so one left, sexual selection. Sexual selection can be described as describing the phenomena that are associated when individuals with certain heritable traits are more likely to obtain mates. So this is still an adaptation for heritable traits, just as all the other forms of natural selection are, but specifically these traits involve mating, or ability to likelihood of obtaining mating. And so this is given a different name for that particular reason. Sexual selection is a little bit more complicated because the kinds of phenomena that one sees that can be described as sexual selection fall in a couple of different categories as shown here. So the two most common patterns that are seen are that there is choice made by females in terms of which male they're going to mate with. This is sometimes referred to as intersexual selection because it involves both sexes, both males and females, because females are choosing males. The other common phenomenon that is observed in sexual selection is competition between males, and so this is sometimes described as intrasexual or sexual selection because it just involves one sex, just the males. That's where these come from. These terms come from. So why is it that we have these two listed and we don't have, say, male choice of females to mate with or female-female competition? Why is it that these are the ones that are listed here? Well, this is driven by a basic asymmetry in male and female sexual reproduction. And so females make eggs, males make sperm. Eggs are, females make larger gametes and fewer of them. Males make large numbers of smaller gametes. So this is a real asymmetry in terms of the investment of the males and females in sexual reproduction. And in fact, there are some cases, so this is driven by the larger investment of females typically in sexual reproduction. There are counterexamples of this, and so the examples when one does see the opposite of what is shown here is usually driven by those unusual cases in which for that particular species, there is in fact a large investment of males in sexual reproduction. And when that occurs in species, then that can cause male choice, for example. But in almost all cases, this is the pattern that one sees. So let's go through the example of male-male competition, which is in your textbook, which involves elephant seals. You've actually seen an example of male-male competition earlier, and that was the giraffes. Remember a discussion of what is the adaptive value of a long neck, and we talked about males that would be fighting fighting using their necks and swinging their heads and necks at each other, and that was related to mating, getting access to females for mating. Well, male elephant seals also interact, and this somewhat gory picture, these seals do not have red fur. This is actually blood, so this is a really sort of violent altercation going on here. Here's a huge cut that you can see right above the water level here. So these individuals can injure each other as they're competing, and so they compete for access then to the females, and the males that are unsuccessful or then move away and do not then mate with the females. And only a very small fraction then of the males, the ones that actually win these competitions, then go ahead and mate with the females. And so this leads to a very skewed representation of the males in the population. You can graph this as your textbook has done, and this is what the graph looks like. The x-axis here is lifetime reproductive success, so this is number of offspring weaned for different males, and the y-axis here is the percentage of males, so down here is 0 at the bottom and 100% up here. So how to read this is, for example, this looks like about 85% or so. 85% of the males have 0 offspring that make it to weaning. So that tells you that the huge, most of the males in the elephant seal populations then are not going to father any offspring. Their genes are not being passed on. There are a small number of seals that end up with up to 10 or up to 20 offspring. There are even a few with 50. And look at this. There are some individuals that have an extraordinary number of offspring that they wean. And so, of course, this means that those particular individuals then are disproportionately represented genetically in the subsequent generation here. So it's just a handful. Females, completely different because of different reproductive physiology, right? These are mammals, and so the females are carrying the young and are supplying milk to the young, and so this is really very different. So the male can go ahead and mate with a number of different individuals, but a female is slowed down in terms of her production of offspring by the rate of mammalian sexual reproduction. So this is now the graph showing what's going on for the females. The y-axis is still offspring, but now this is thinking from the perspective of the individual females instead of the males. The y-axis is percentage of males, going from percentage of females, 0 at the bottom, and then just up to 75 here. You can see that just with the males, there are in fact some females that never have any offspring. This will happen in any kind of natural population. But there are no females way out here having extraordinary numbers just because, of course, they're limited by their reproductive physiology. So completely different pattern between the males and the females in the elephant seals. Now, this kind of pattern is seen in some species. I thought it might be interesting to show you something that's sometimes called the Genghis Khan effect, which is a example of disproportionate genetic representation in a human population. You may or may not remember from history that Genghis Khan was the founder of the Mongol Empire, and so many, many hundreds of years ago, the Mongol Empire stretched over a very large fraction of the globe. Here is a map from showing where the extent of the Mongol Empire was in the year 1279, for example, and human population geneticists have gone into this area and done analyses of the genetic makeup of the individuals in this area and have found that a fairly large percentage of all males in this area, namely about 8% of males in this particular area, are thought to show a lineage that can be directly traced back to Genghis Khan and his sons and so on. So this is really from quite a long time ago. You can still see the signal in those populations. Okay, so let's leave natural selection now and move on to the next evolutionary mechanism, and that is genetic drift. This is one that students sometimes find much more confusing because it's the argument deals with probability and understanding the role of chance when you have a very small numbers. So genetic drift is defined as changes in allele frequency which are just caused by chance fluctuations. So there's nothing adaptive about a particular allele that then is more represented in the population. It's just chance events, and chance events are much more likely to happen if you have a small sample of small population size, which is why this is particularly pronounced at small population sizes, and with very large population sizes it is negligible. So if we consider this case where each oval represents an allele in the population, we've got a little tiny population here. This is 4 individuals. They're diploid, which is why we have then 8 alleles as shown here. And so as these 4 individuals then reproduce, and we look at the next generation, and it should not surprise you, it probably makes intuitive sense that just by chance we might end up with not exactly 25% green alleles and not exactly 75% blue alleles, which is what we have in this little starting population right here. Now there are a couple of different phenomena in biology that are examples of genetic drift that are given specific names because they're common and interesting in populations, and those are founder effects and population bottlenecks. And what do these have to do with genetic drift? Well, a so-called founder effect is a name given to a case where you have some habitat, and so I just drew a little island, and there are no representatives of some particular species on this island, for example. And then a couple of founding individuals show up. It could be a single pregnant female, or it could be a small group of birds, or a couple of lizards clinging to a log. Something washes up on this island or flies to this island, ends up in this island, and it's a small number of individuals. Under those circumstances, then, you're starting with a small population. This is a name given for the kind of changes that one would expect on this newly colonized area, just simply because of small population size. Population bottlenecks is something a bit different. This is what happens when you have a large population. So if this is population size here, and then something cataclysmic happens. There's some awful geological event. Volcanoes go off, or earthquakes, or some epidemic races through, and the number of individuals in this population crashes down to some small number. They don't go locally extinct, they're just down to a small number. Then so this is then given the name bottleneck, because if you graph it like this, it looks a little bit like the neck of a bottle. So under these circumstances, then, it should make sense to you why we suddenly now have a small population size, and at this point then, generation after generation, while we have this small population size, we're more likely to see for genetic drift to be a very important cause of changes in allele frequency. These population bottlenecks are of concern in conservation efforts, where one can have suddenly a very smaller population, which usually, of course, means that there's much less genetic variation in that little tiny population, just because there's fewer individuals with alleles in that population. One can get an idea of the magnitude of changes in allele frequency with population size by doing computer simulations. And this is a graph from your book. And so we're going to go through two different computer simulations, one in which the population is 4, so this is a very small population, and one in which the population is 400, or a larger population. For this computer simulation, we're just considering the very simple case of one gene, 2 alleles, A1 and A2. And what we're looking at on the graph here is time on the x-axis, or generation. And on the y-axis, we have frequency of the allele A1. And of course, this then also tells us what's going on with A2, because they add up to 100%, or a frequency of 1. Now, for all these genetic computer simulations, we're going to start with half of each one, right? So we'll start with 50% A1 and 50% A2. The y-axis is frequency of A1, so we're just starting right here at 0.5. What this graph shows you is that a number of different simulations were run, because of course not exactly the same thing happens every time, because chance is, after all, really important. So sometimes it goes down, like this one, and then it goes back up, and then it goes down again. Sometimes they go up. So this shows 1, 2, 3, 4, 5, 6, 7, 8, 9, on the order of 9 or so different simulations showing the characteristic behavior that one sees just by chance. The population, by the way, starts at 4 and is kept at 4. So each generation has a new 4 individuals, and we're looking at the alleles in there. Now notice that in these simulations then, by the time we hit about generation 20, we have either hit 100% or 0% for A1, right? So the way of describing that is that the allele was lost or it went to fixation. So for example, at this point right here, then we have A1 is 100%, which means, of course, A2 is 0%. So A2 was lost. A1, we would say, went to fixation. In this particular example here, A1 went to 0. Right, so there's no more A1 in this population, simulated population. A2 is now at 100%, so this one went to fixation, and this one was lost. And notice that because we don't have any way of replacing A2 or A1 once the other one is lost, once you hit a fixation or complete loss of an allele in a simulation like this, you just stay at this forever, to infinity, right? Because where's the other allele going to come from, right? So what this tells us is in this population size, then one can then estimate the probability of losing one of 2 alleles when you start with 50% representation of each, and the answer for this is in about 20 generations. So again, this is another concern for, say, conservationists who are worried about little tiny populations where the genetic variation is already much smaller, thinking about how as time goes on, if the population remains small, is there going to be more loss of alleles in the population? Completely different for a larger population size. Here we have our population size of 400. And again, it's 400 next generation. We're still assuming 400 and so on. So the population is not growing. It's different individuals each generation. But notice that these are going up and down, but the magnitude of the wiggle is much more reduced The generation by generation changes in allele frequency are much smaller. This is because genetic drift, which is driving those random changes, we still have random changes, right? It doesn't look like this, right? It doesn't look like that. We're still getting random changes. That gives you an idea of the magnitude of genetic drift then, the up and down movements, the changes that are just due to random events. But notice that 100 generations have gone by and we haven't lost our alleles in these simulations. Again, doing a large number of simulations. Gene flow, our next mechanism. Gene flow is defined as a change in allele frequency in a population that is due to immigration or emigration, right? So if an individual leaves one population, and now this little cartoon in front of you is supposed to represent two populations. So we've got one population over here on the left, and we've got a second population over here on the right. And it's the same species, and we just have a very simple issue where we have two alleles, and they're either green or they're blue. Notice we have a slightly different allele frequency on the left in population 1 than we do on the right in population 2. We've got a higher proportion of green alleles over there on the left. Now, each oval is representing an allele in the population, so this is like a gene pool kind of representation or allele. Just looking at the alleles, not worrying about where the individual is. So why would an individual moving from one population to another change the allele frequency? Well, let's see one move. Here we go. Oh, there's the individual and it went, right? And it's diploid, so it had two alleles and it took it with them, right? So notice that both the allele frequency in the population from which it left and the population to which it moved have now both experienced a change in allele frequency because this was a heterozygote. So now the allele frequency of green alleles went up on the right and down on the left. So the end result or consequences of gene flow, movement between populations, is that the populations become more similar to each other genetically. And that just sort of makes sense to you. If you mix things, they become more homogeneous. The more gene flow you have between populations, the more similar they will become with each other. So gene flow makes things more similar. Students sometimes think that this is gene flow. If an individual just goes off. No, this is not gene flow, okay? Right? Because here we had a little habitat where there was nobody. We just had a founder effect, right? We just had a little founding of this population. Gene flow specifically means going from one population to another, not going and colonizing a new area. This is not gene flow. It's just an individual going off and colonizing something completely different. Okay, final evolutionary mechanism here, mutation. So this is, of course, any permanent change in an organism's DNA, and there are many different kinds of mutations. It can be a very tiny change, such as a point mutation, where a single nucleotide is modified, or it can be much larger. Mutations are usually deleterious. This should make sense to you that just a random change of something that's working okay wouldn't necessarily be beneficial. Certainly, mutations sometimes can be beneficial, but most of the time they are deleterious. They're at a pretty slow rate. So overall, although mutation is ultimately the source of all genetic variation, it is slow enough that it is usually much lower in magnitude than other evolutionary mechanisms that are operating in populations, such as natural selection and so on. So here's an example of how slow the rate is. Of course, it depends on what organisms you're looking at and what stretch of the DNA and so on. If one looks at the maximum rate in gametes, for example, typical rate that's reported is about 1 out of 10,000. And now, in my remaining minute, I'm going to end with a clicker question. It hasn't clicked yet, so I'm going to go ahead and do this. It says 349 down here. Consider this. This is relatively simple. I expect 100% on this. We'll see. Consider a group of dogs, and in fact, the Afghan hounds specifically, there's a picture of one. They're all descended from a very small group of dogs that were taken from Afghanistan to Scotland in the 1920s. So a little tiny group of dogs. And then later on, some of those descendants were brought to the US later. Would you, you should finish the filling in this sentence here with these blanks. So the first part before the comma, you would put in the first blank and the second part here. How would you expect today's Afghan hounds in the US to be related to, or to be related to the Afghan hounds still in Afghanistan? Would you expect them to be the same as or different from? And because of what? Would you expect them to be the same as Afghan hounds still in Afghanistan because of mutation, the same as hounds of Afghanistan because of genetic drift, etc.? You may assume populations in the US and Afghanistan have remained completely separated since the small group of dogs was brought to Scotland. Okay? Yes, you should be chatting with your neighbor, compare notes, click in. I'll give you another 15 seconds or so to figure this out. Okay, as soon as this stabilizes, I'll show it to you. Okay, looks like it has stabilized. Let's see if you guys got this right. Whoops. Got a last few. Okay, everybody clicked in? And 10. Whoa. We'll revisit this one on Friday before we talk about speciation. Okay, I'll see you on Friday.